Hey, hello, everybody. I'm Jenny Hathaway. I've been working with Diana at Northup for a long time, and today I'm going to talk about the wild and wonderful caves of Great Basin National Park, as well as the Humboldt-Toiyabe National Forest in Nevada. So I like to start with my acknowledgments because this work is done by a whole bunch of people, not just myself. This work was funded by the Park Service and the Forest Service, and so I have Joe Medley and Shiloh McCullum who did field help on our two different field trips. Ryan Ozatalar and Elizabeth Walker did both field and lab work. Evan Bowen came with us in 2025, and he is doing work with sulfur isotopes out of Dan Jones's lab down at Tech. Mike Spilde with the SEM, and then a whole host of Park Service people. This project was really spearheaded by Gretchen Baker and Doug Powell, Aaron Levesque, Victoria Griever, and Olivia Ford. And then Peter Dresky, Jessica Sisu, Leila Mende for field support, and Kenneth for always the beautiful photos as well as field support. So where are we? We are in Nevada. We are in White Pine County, which is way over on the eastern side. And so we are mostly here in Baker, which is the gateway to Great Basin National Park, if you've never been. The park is mostly here in the Southern Snake Range. The Forest Service caves are mostly up here in the Northern Snake Range, except for we have one cave that's over here in the Egan Mountains. Ely is the nearest town. Baker is a town of like 100 people. There is nothing, nothing there. But Ely is where you have a grocery store and some more things. There is a wonderful general store that does pizza night on Wednesday. Listen. Okay, our project summary. We are part of a wild cave project that was spearheaded by Gretchen and Doug to survey the caves of the regions. They did geology work, invertebrate work, hydrology work, all sorts of other things. I included a few little inverts for any invert people out there. I'm a microbial person, so I don't know what those are, but they were fun to see. We went to about 10 caves, 5 park caves, 5 Forest Service caves over a 2 years period, and we visited most caves twice. And we were collecting samples for bulk chemistry, for DNA analysis, for SEM work, and the sulfur isotope analysis done at New Mexico Tech. So the types of caves mostly are suspected hypogean caves. Here's a couple of the lovely vistas within the cave. This thing in the center here, it's probably about 40 feet tall, really cool. Lots of these boneyards. I've done vast majority of my work in lava caves, so these are like totally different to me. And so they were very highly decorated and a lot of fun to be in. We did have 2 fracture caves and one cave that's like fracture and a bunch of other cave formation types. Both the main fracture caves were up at elevation on top of Mount Washington at 10,000 feet. The other caves are like 6,000 to 7,000 feet. And then finally, we have the show cave. The show cave is just Lehman's Cave. It's the crown jewel of Great Basin. It's highly decorated, really fun to go to. And we got to go behind the scenes as well to the part that is not lit for the public and do some work back there as well. Oh yeah, here we are, going down into the back rooms. So what we did, we drove. Baker being in the middle of nowhere, everything is several hours away. The ones on top of Mount Washington are 3 hours of switchbacks. So glad I was not driving. But we drove and then we hiked. We hiked, some of them were like 3 hours hike after driving. And then finally we got to go into the caves. And this is Leila Mende descending into one of the alpine caves. What we did once we're in the caves is we looked and we looked and we looked. And so here's Elizabeth Walker and Joe Medley, 2 of the people. I've got Jessica, Victoria, and Olivia, and Evan here. So we looked, we took notes, and we finally decided what we wanted to sample. And then once we'd done that, we went ahead and sampled. We collected the DNA samples aseptically. So we wear gloves, we flame sterilize our tools, we try and make sure none of our human DNA is getting in with the cave stuff. One thing we really worked on this trip was to try and describe the color with paint chips or the Munsell color chart that I supplemented with a lot of paint chips because they just didn't have the variety of colors that we were seeing in these caves. And then we took data like air temperature, humidity, wind speed. And Evan had a light meter for a few of the caves where you could still see twilight from our sample sites. What we did once we were back in Albuquerque is we took our samples and they either went for SEM work with Mike and Diana, or what I did along with Ryan and Elizabeth is we extracted the DNA, sent the DNA off for sequencing, and then we sit in front of the computer for a while and try and make graphs. And then we look at the graphs and try and understand what they all mean. So that's what we do here back in Albuquerque. So a couple of the vocabs that we use, archaea and bacteria. So this is like a tree of life. And so bacteria, oops, wrong button, hold on. Bacteria are highly diverse. They live everywhere. They do everything. They're fascinating. And you can see there is a lot of different types of bacteria out there. Archaea are considered an older life form. They really like extreme environments, but we're finding them in places such as caves. They're thought of being like the base of the tree of life. And we're finding that they're really important in doing nutrient cycling, especially nitrogen. And so these are archaea. And then everything else that we think of as living is in this little branch down here. Well, we're somewhere down here. I don't know. I haven't looked to see which tiny branch we are. But the other thing I want you to notice is that in 2004, we knew about the stuff that's blue on this figure. And in the last 20 years, all those other colors are parts or groups of bacteria that we have discovered, or groups of archaea. So a lot has happened in the last 20 years in terms of our knowledge of the microbial life. So I'm going to take you on a little tour of what's in the caves. This is Gretchen Baker here and Joe Medley. You can see we go through all lengths to get our samples, lying all in all sorts of fun places. So in the caves, there's moon milk. And for me, coming from lava caves, I've never seen these giant walls of moon milk that are, 40 feet long. I've never seen so much moon milk. But there's moon milk all over the place. Slightly different in color variation, which is why that color identifying was really important to us. And so we took a variety of samples of moon milk. And when we look at them under SEM, yay, we find life. We're always so excited when we find signs of life. So we have these features and they have little filaments on them, or these kind of fuzzy looking sausages. As Diana says, we're not really sure what any specific form is, but we'd like to see the same kinds of features in samples. So we saw a lot of variation between the samples of moon milk, both in the SEM and in the bacteria and archaea. The bacteria had a really large diversity. It had a lot of unnamed or candidate phyla, so stuff we know very little about. We maybe only know about them through their DNA sequences. But we had one that I wanted to point out. It was called a Zixi bacteria. It's been found in other moon milk in Europe, as well as a variety of other things, and it is a sulfate reducer. So it might be very important in Dan Jones's kind of work to see what he's finding about sulfate isotopes in relation to where we find these sulfate reducing bacteria. In the archaea, we found a lot of, again, unnamed, unknown candidate or genera, as well as ones involved in nitrogen cycling. Okay, so now we're going to take a journey of discovery, but really it's a journey of Discovery Cave. So Discovery Cave is this really highly decorated with lots of unusual features in it, one of which is the folia. And they formed at the water table as the cave was forming, and it's just this wall of these fun little shelves of folia. Really fun to see. Here's an up closer picture. You can kind of see what they look like up close. When we look at them under the SEM, we see calcite crystals as well as sort of bacterial features that we have seen in other locations. In terms of the bacteria, we had, try but not butcher this name, a Haliangium bacteria, which produces antifungals, and then a lot of nitrogen cycling and uncultured. In the archaea, there was only two types. One was a nitrogen cycling and one was the salt loving. So salt loving and nitrogen cycling in terms of our archaea in these samples. Continuing in discovery, we had quills. I don't know if anyone's ever seen them before. They were amazing. They looked like this starburst of little or big crystals coming out of sort of in a spherical pattern. There's a whole little alcove just chock full of them, as well as some other places in the caves. And in the quills, we found an interesting form of calcite over here. So just some different mineral variations that are not as frequently seen in our samples, as well as some signs of life. There were fewer types of bacteria in the quills than the moon milk, but a lot of them are unknown. You're going to hear me say that all the time. We found unknowns and we found nitrogen cycling things. The archaea, there were only 2 genera, and they were both involved in nitrogen cycling. Okay, we're going to continue our journey of discovery. One thing that Doug pointed out to us on our second trip was these sort of smaller quill kind of formations that seem to be being enveloped in by minerals and making those smoother formations. So that would be a really interesting place to study over time to see how these features are changing and if they are in fact morphing into other features. Okay. What else is in the cave? Amber rat. Who knows what amber rat is? A few people. Amber rat is the mineralized urine of pack rats. And they have huge, extensive formations in these caves of this. Here we have some more up-close pictures of them. We haven't analyzed the SEM of this particular sample type, but we did do bacterial data on them, and they are unsurprisingly salt lovers and nitrogen cyclers. For both the archaea and the bacteria. Coralloids, we have lots of different examples of coralloids in these samples, or in these caves, and we collected them in almost every cave we were in. Here, we kind of have a broad category of coralloids. We include sort of popcorn and polyps in this broader category of coralloid-like structures. When we look at them in the SEM, we have, again, some calcite, but lots of signs of life. So we have, this is a really interesting shape that caught Diana and Mike's eye. Not sure what it is, but we also have lots of filamentous things that look biological. The coralloids, again, have a lot of different types of bacteria and a couple of different ones that are thought to be involved in carbon cycling, including Procella and RB41. And in the archaea, we had novel nitrogen cycling and salt-loving archaea. My last big category that I'm going to show you today is mineral crusts. There are a lot of different types and colors of mineral crust all over these caves. The picture's not coming out very well in this one. It is a pale pink mineral crust that was in little patches all over, especially the alpine caves. So that was really fun to see. Really hard to get off the wall. So that took a lot of work. But we have brown mineral crust, white mineral crust, yellow mineral crust, soft crust, hard crust, anything you can imagine. We found in these caves. And again, they were highly diverse in their types of bacteria. Our favorite beads on a string, we found those in these types of samples, as well as other filaments and other kind of bacterial type things. So the main takeaway from the bacteria and the archaea was there was a lot of variability in the different mineral crusts, which you would expect because they don't all look the same. So there's a lot of variability across mineral crusts. Okay, so now we're going to get into a little more hard science. We're going to ask a couple of questions. So the first question is, are there different types in the number of types of bacteria or microbes between the caves? The next series of slides all are going to have these 2 graphs. The first one is kind of our raw count, the species richness. And then based on the amount of like unique types of bacteria or archaea in there, we can do some math and come up with an actual estimate of the real diversity as opposed to what we just saw. And so that's with Shannon. So our species richness with terms of the archaea, you can see Liberty Cave is highly, highly diverse in terms of the archaea. Liberty Cave is the one cave over in the Egan Range. So it's in a different mountain range than all the other ones. Otherwise, we saw sort of, you know, a variety, but everybody was kind of close, except for Lehman's. Lehman's, the show cave, not a lot of archaea my guess is that the human impact is letting the bacteria outcompete the archaea in that environment. So next we have the bacterial side. I left the archaea over here so you can kind of have a comparison. Liberty is no longer the most diverse. Actually, Lehman's is way up there, along with this other cave here, cave NS18. But again, we can see a lot of variability between the caves. But this just looks at the types of bacteria. We're not taking into account the amounts. We'll get to that in a minute. So we talked about how they differ between caves. How about how they differ between sample types? So again, with the archaea, we have all our different sample types. You can see some of them, we only had one instance of those, so they don't make very good data points, but I included them because you can see maybe that's boxwork. Maybe we should go try and find some more boxwork and do a little bit more work on it because it seems to have a lot of different archaea in it. The calcite and the cave silt, they had, and we got that with our DNA too. It was really hard to get DNA out of them, and they did not have a very big diversity. In terms of the bacteria, again, we're pretty even across our samples with some variation, but nothing stands out as like, this is really unique. So all of our caves have a good amount of diversity in them, or all of our sample types have a good amount of diversity in them, but we don't think that they're really the same as each other, which we can see in our next slides. So this is, what if we look at how similar the community of microbes are across the different caves. And so in the archaea, we have one cave here, oops, button again. One cave here, the snake cave, that has kind of a tighter ellipse around it, this pink one. But all the other ones are kind of all over the place when we look at them by cave. But if we look at the bacterial side, one thing I noticed is that we got 3 caves over here all sort of right on top of each other. And if we look at them by cave type, we can see all 3 of those caves are fracture caves. So the 2 alpine caves and the 1 cave ice cave, which is at a lower elevation, but is also a fracture, and then it has some secondary cave formation types in it. But it's primarily a fracture cave. And actually in the archaea, we see a similar kind of pattern with all of our 3 fracture caves joined together in one area, and then all the other caves, all the hypogean caves in a separate circle. And then finally, if we look at them by sample type, I don't know, it's kind of a mess. So it's hard to say that that's what is actually driving the differences that we're seeing. So that's where our further work will be. The data I showed you is kind of like a fingerprint, what types are there, but we know nothing about what other genes they have and what are their metabolic capabilities. So we are doing a big 80 sample metagenomic study from these same samples, and we are just working on getting the sequencing done now, but then we can start asking questions like, what possible energy sources are the microbes using? How are the organisms getting their carbon or other essential nutrients? What organisms are actually involved in the different steps of cycling nitrogen? And what can we say about the relationship of the organisms based either by the cave or the speleothem types? So we have a grad student, and that is his spring project, to analyze all this data. And with that, I will take any questions. No questions, woohoo. Yes, Mike.
Is there a lot of variation in temperature?
So yeah, I was thinking about that today. Actually, yes, but I think this between like 7 and 13, if I remember correctly. So not a huge flux. But some caves are definitely warmer than others. Yes.
On your sort of tree,
Uh-huh.
you showed bacteria,
Mm-hmm.
and then you showed archaea,
Oh, wrong way.
or eukaryotes.
Yep.
Way in the beginning.
Way in the
And
beginning.
archaea was up, but it doesn't really speak to that.
Yeah.
I mean, it doesn't really a tree to talk about what's more ancient than.
Yeah. What it shows is, I mean, we believe it's single-celled organisms were the start of life, right? And the archaea are known, like that cave we've talked about in the past, discovered under the house in Santa Fe, had really high radon and a lot of archaea and not many bacteria. So they like these more extreme environments. And so some people would say there are two domains of life. Archaea and eukarya are one of them. And the eukarya came off of the archaea, and then bacteria are another. So this is an area of science that is changing rapidly with more data.
Because before it was always the archaea, so ancient,
Yeah.
but now that's changing.
Well, they still think it's ancient. It's just that the eukarya kind of came off of it, not from like a center point. Awesome. Thank you.